So our next speaker is Cheng Huang, who's going to tell us about the imaging of neural spiking and dendritic dynamics in awake flies.
Okay. Good morning, everybody. My name is Chen. I'm a postdoc from Schnitzer's lab at Stanford University. And I'd like to thank the meeting organizer for giving me this opportunity to present our latest progress on the development of genetic encoded voltage sensor and its application in fly. In order to understand the mechanism of high-speed information processing in the brain, neuroscientists designed a good way that could faithfully and reliably report the cellular and subcellular electrophysiological activity with milliseconds resolution in the brain. And the two current prevalent methods for doing so is in vivo electrophysiology and in vivo calcium imaging. But both of them have their own advantages and disadvantages. For example, the electrophysiology has a very good temporal solution for spike detection and have a very good spike timing accuracy. And it also could detect the very small subthreshold membrane dynamics. But it's really hard to use it to target specific neuron you designed and to record it for a very long time. It's also very hard to recording from small cellular compartment like the dendrite and axon, and or from a large number of neurons altogether. On the other hand, calcium imaging, especially with the genetic encoded indicator like GCAMP, it's very easy to use it to target specific neuron or even the small compartment dendrite axon. And you can also do the population recording at the same time. But the disadvantage is it suffers from its poor temporal resolution, and also the calcium is not a direct indicator of the neural activity after all. So in principle, the ideal way is to have a very good voltage sensor that could potentially combine all the benefits together. But unfortunately, we haven't had it yet. But indeed, in the last few years, this field have made a huge progress on the sensor development, and I'd like to update with our newest version that could report the individual action potential and also the spiking trend in a live animal, particularly in fly. So our lab previously introduced a voltage sensor design configuration called FRET-opsin. In this configuration, we combine our voltage sensor domain from a rhodopsin and also our bright fluorescent. The rhodopsin could function as a photon acceptor, and the fluorescent is a donor. So when the membrane potential is depolarized, the rhodopsin actually becomes a better photon absorber, so the fluorescence you can detect is decreased. That's why you can see the scale bar in all my experiment is actually reversed. Our first version called MAQ-M-Citron, and this version has a fast kinetics than the previous voltage sensor like ArcLight. And it could report the single action potential in culture neuron or even brain slice. But unfortunately, the single noise ratio is not good enough for us to detect the action potential in vivo. So we improved the sensor in three ways. The first, we changed the rhodopsin to another kind called ACE, and this rhodopsin have an even faster kinetics. Second, we changed the fluorescent to our brightest fluorescent we could find, that called M-neon. And the third, the absorption and emission spectrum of the rhodopsin and the fluorescent is better overlapped, so we could have a better energy transfer efficiency between them. As we expected, this sensor has even faster kinetics than the previous one and other fast voltage sensor like ASAP. And it could reliably report the waveform of single action potential in culture neuron and brain slice. And more importantly, for the first time, we could we could detect the single action potential in vivo. In this case, it's in the mouse cortical neuron. So we introduced this sensor into fly brain. And first, we characterized the performance of this sensor in a olfactory local neuron by using a simultaneous patch clamp and optical recording on an isolated brain preparation. We can see the sensor could report the action potential nicely, and you can even see the sub-threshold fluctuation. And in a more extreme condition, we could introduce a very fast spiking trend, and this sensor could report every individual spike in this fast spiking trend. And by doing so, we collected over 18,000 spikes, and the sensor didn't miss any of them. And by comparing the spike timing from the optical traces and the electrical traces, we found that actually the spike timing accuracy is around 0.2 milliseconds. It's quite low and is approaching the theoretical limits of the optical detection of action potential. So next, we moved to the in vivo experiment. And we found also in this local neuron, we could see the odor-induced spiking and also the spontaneous spiking in a single trial experiment. And we can calculate the firing rates of both induced and spontaneous, which provide us a better way to quantify the neuron activity than just comparing the difference in the fluorescent amplitude. And also, this sensor could report the excitation, induced excitation, and also the induced inhibition, which is harder to be detected by calcium imaging. We moved to try to see if the sensor could detect dendritic dynamics, and we imaged the dendrites of a projection neuron, which labeled the specific glomeruli. And we can see the odor-induced dendritic dynamics with very detailed information, which could not be captured by the calcium imaging also. And it also can report excitation and inhibition. So besides the somatic spiking and dendritic dynamics, the fast imaging could allow us to visualize some very interesting intracellular property. The first example is the submillisecond-scale dynamics of spike propagation. So in this case, we could see the spike is initiated in the cell body of this local neuron, and it is propagated from the right side to the distal process on the left side. And by averaging many action potential together, we can actually capture the sub-milliseconds delay of those three regions. And in the second cases, we could image the dendrite and axon region together, and we can see the dendrite and axon have a largely similar response, but except they have a 3 milliseconds delay. So that's the first time we can actually calculate and capture the signal propagation speed in vivo in fly brain. In the third example, we actually imaged the dendrites of projection neuron on two hemispheres of the brain. And you can see the response from the two hemispheres is largely similar, but it still has some asymmetric response, as here you can see, and here and here. So this allows some people, if interested in the lateralization of sensory information, they can directly record those differences from two hemispheres and to understand how those lateralization could guide the directional movement of the animal behavior. So in summary, I showed that our sensor could reliably report the spike in fly brain with a 0.2 millisecond spike timing accuracy. And it could also record the dendritic voltage dynamic and some interesting intracellular phenomenon like the voltage propagation delays. And we're currently developing new version of sensor that could have improved single noise ratio, and also we're trying to combine the optogenetics with the voltage imaging to achieve the optical electric physiology. So besides our lab, actually there are several labs, they also introduce their own version of voltage imaging into FLI, and they have different aspects of applications. So I think the take-home message I want to leave here is that it's actually a very good time to start thinking about what kind of new experiment that could combine the wonderful genetics toolkits in FLI and the emergent voltage imaging technique to understand the genetic basis of neuronal processing. So I'd like to thank our voltage imaging team at Stanford, especially my mentor, Max Schnitzer, and my colleague, Yang Gong, who's the designer of this sensor. And also, I think I should thank FurFlies, who devoted their life to my work, and also the funding agency who supported our work. So thank you for your attention. I'd like to take questions.
Some very cool work here. So I wonder, have you compared side by side the recent developed ASAP 1 or 2 in flies with your sensor?
Actually, I didn't compare them side by side, but I tried ASAP personally. So we found the problem with, first, we're using different imaging technique. For our experiment, we used the 1 photon faster speed imaging, and we could see the single spike. But with ASAP, the fluorescence brightness is not good enough for detection under the kilohertz imaging rates. But their advantages, they could be used under 2 photon, but our sensor is not able to use under 2 photon right now. So I think we have different disadvantages and advantages.
Thank you.
Okay, thank you very much.